Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show, bringing you another really fascinating guest today, helping to create a better and safer tomorrow for all of us. Today we have the honor of being joined by Dr. David Markowitz, who is a program manager at the Intelligence Advanced Research Projects Activity, also known as IARPA, which is an organization that invests in high-risk, high-payoff research programs to tackle some of the most difficult challenges of the agencies and disciplines of the United States intelligence community. Their mission is to push boundaries of science, help develop solutions that empower the U.S. intelligence community to do its work better and more efficiently for national security. They don't specifically have an operational mission. They don't directly employ technologies to the field, but they facilitate the transition of the various research results to different customers in the intelligence community for operational application. And they work across a broad range of technical areas, including mathematics, computer sciences, physics, chemistry, and biology, as well as neuroscience, linguistics, and cognitive psychology. Currently, Dr. Markowitz leads 3 research programs that exist at the intersection between biology, engineering, and computing. These are the FELIX program, which is involved in revolutionizing the area of biosurveillance, MIST, which is developing compact and inexpensive DNA data storage devices, and MICrONS, which is involved in guiding the next generation of machine learning algorithms based on computations performed in the mammalian neocortex. Previously, as a researcher in neurosciences, Dr. Markowitz published first author papers on neural computation, the neural circuit basis of cognition in primates, and neural decoding strategies for brain-machine interfaces. He received his PhD in molecular biology and neuroscience from Princeton. He did his bachelor's degree in management science from MIT, and he was a Department of Energy computational science graduate fellow and a Schwartz Foundation postdoc fellow in computational neuroscience. Dr. Markowitz is also a recipient of the 2019 Presidential Early Career Award for Scientists and Engineers. We're honored to have him with us today. A lot of very exciting topics to get into. Dr. David Markowitz, welcome to our show.
Thank you, Ira. It's really a pleasure to be here with you and with your listeners, and I'm grateful for the opportunity.
It's really great to having you, David. I'd love to start off as we typically do. I'd like to hand you the floor for a little bit, just to talk a little bit more about you. If you could take us a little into your background, everything from where you grew up, the development of, what I'll go back to continually during the show, these really convergent interests in molecular biology, engineering, neuroscience, and computing, and a little bit of sort of what sparked the beginning of this fascinating journey you've been on. I think that'd be a great way to start things off.
Sure. Well, I'll start by giving you a brief overview. I'm a biotechnologist and what I would call a scientific entrepreneur, and I'll explain what that means in a moment. For the better part of the last decade, I've had the privilege to create and actively manage a large portfolio of applied research and development programs at IARPA, which, as you mentioned, is the high-risk, high-payoff research arm of the U.S. intelligence community. IARPA's mission is to deliver transformational capabilities for national security, and the agency is uniquely empowered to do that by applying significant resources to take big swings at the hardest problems. Now, my job as an IARPA program manager has been to practice scientific entrepreneurship, which is, for each program, my job is to define a major customer pain point, to articulate a vision for how we can address that problem by developing new technologies, to convince the government that it's worth giving me a big pile of money to go and make this happen, and then to execute a principled strategy for delivering impact to customers. Speaking as someone who was formally trained in entrepreneurship at the MIT Sloan School of Management, running programs at IARPA is not that different from entrepreneurship in industry, except my investors, my board, and my customers are all members of the national security community. We have some economic ways of measuring the impact of our work, but we also measure success operationally and sometimes even in terms of lives saved. And that's been really satisfying for me personally during my time in public service. Lastly, I'll mention that the overarching theme of my career has been bridging biology and engineering disciplines to solve problems at unprecedented scale. I've been fortunate to help develop new devices, diagnostics, and detection assays. I've developed machine learning systems that accelerate discovery, and I've delivered tools that are now in widespread use by customers around the world. So to tell you a little bit about my journey, ever since I was a kid, I've been fascinated by finding new and different ways to solve problems. In high school, I actually used to get in trouble in calculus class by developing various tricks to solve problems on exams with less work than the teacher wanted us to show. I would lose points for doing things correctly, but not the prescribed way. When I got to college at MIT, I loved science. I knew I wanted to be a scientist. But I found myself less interested in the standard curriculum of biology and computer science and more in what new methods could allow us to accelerate discovery or solve really hard problems at scale. I just wasn't a good fit for the vanilla undergraduate science curriculum. Frankly, I was impatient to do research and build stuff. So I chose the one major at MIT that was all about the science of developing new ways to solve complex problems with multidisciplinary teams, and that was management science. In parallel with my undergraduate work, I immersed myself in real-world problems having to do with large-scale data management and analysis. So I did undergraduate research on data aggregation in computer science, and my work was spun out into a startup company that was subsequently acquired. I also did consulting work for a range of companies in the US and the UK who were trying to use massive data sets to solve problems. I did that throughout college and for about a year or so after college. And when I was ready to pursue my scientific training, I looked for a field where I could solve real-world problems for people in need. But the only way to do it was by bridging disciplines and operating at scale, which is that particular itch that I always wanted to scratch in college. And these are things that appeal to my natural desire to solve the hardest problems, I guess. And I quickly fell in love with neuroscience. The challenge of neuroscience is that everything is important at every scale. I mean, you can make a big impact defining a specific niche, working at the molecular scale only, but changes at the molecular scale in the brain can have profound consequences for circuit function at the level of billions of neurons, leading to changes in cognition and behavior. So I made it my mission to train at every level. I did a PhD in molecular biology and neuroscience, as you mentioned. I trained as a computational scientist through a fellowship with the Department of Energy, which exposed me to the tools of high-performance computing applied to biology for the first time. And I practiced the mechanics of quantitative modeling of biological systems at every level, from genetic regulatory circuits all the way to large-scale neural networks. After graduate school, I pursued postdoctoral training that really focused my skills on translational impact for people in the real world. In neuroscience, if you want to have impact for people with movement disorders or psychiatric or cognitive disorders, you need to be able to pin down exactly which systems are failing, and you need tools for targeted intervention. So I did a postdoc at New York University in primate systems neuroscience and neural engineering. While there, I developed a new large-scale chronic recording technology, chronic meaning you put it in for a long period, that allows you to move lots of electrodes once they're in the brain. And this allowed me to systematically map the responses of neurons throughout a three-dimensional volume of the brain, from the top of the cortex, which is the outermost part of your brain. It's the evolutionarily newest part, and we think it's the part that allows humans to be uniquely smart amongst the animal kingdom. So we could move electrodes from the top of the brain all the way down to the bottom layers of cortex. So you can sample in a dense three-dimensional volume and really get the first complete picture of what neurons are doing throughout the, a 3D volume of cortex. No one had ever done that before. So I used those data to identify optimal strategies for decoding movement plans from the brain, which layers have the most information, which frequency bands, which electrode configurations, et cetera. And that work has since helped to make brain-machine interface systems more robust. You don't need to retrain them as often. So that's satisfying for me to see my work have some small impact that may ultimately help patients at scale. I also use that recording technology to determine that different networks in cortex support different cognitive processes that are linked with disease, schizophrenia, and other disorders.
Yep.
And that work suggests that we may need very highly targeted approaches for future disease interventions. Instead of bathing the entire brain in a drug, which is the current pharmacological approach, we may need to target specific populations of neurons that support just one cognitive process that's failing. And honestly, that work highlighted for me how little we still know about the structure and the function of networks that support cognition in the brain. And I realized that absent some fundamental advance in our ability to study and target and manipulate neural networks, it might be decades before I could deliver significant impact for patients. That was a little bit discouraging for me. I mean, I was planning to put in the decades of work, but I had hoped to have impact sooner. So it was around this time that the US government launched the BRAIN Initiative.
Okay.
It is now a $5 billion plus effort to develop new tools and technologies that accelerate our understanding of the brain and allow us to deliver more impactful therapies for patients in the future. I was approached by IARPA during my postdoc to help design and lead what was then the neural circuit mapping component of the BRAIN Initiative. And I jumped at the opportunity. I dropped everything and I said, sign me up. This was a once-in-a-lifetime opportunity to drive massive advances toward new capabilities that we need to understand disease and ultimately deliver impact for patients. Now, IARPA's goal, which is my goal as well, was to use neural circuit maps to guide advances in machine learning and neuromorphic computing, which have specific utility for national security. But these tools are dual use. Like you can use them to study disease or computing in the brain. And that's been a theme of my work, is developing advances at the interface between biology and engineering that have diverse applications for national security as well as biomedical needs. And so that's the story of how I came to IARPA. From there, I was like a kid in a candy store. It's such an empowering place for highly motivated people with great ideas for applied research. And my portfolio quickly expanded to encompass programs in neuroscience, synthetic biology, biodetection, and other areas. And nearly every technology I've developed at IARPA ultimately creates opportunities to advance neuroscience, but they're all principally focused on solving specific national security problems. So that is my brief biography and explanation of where I am today.
Yeah, that's an awesome intro, David. And I just, for the audience, I recommend everyone that's interested in sort of reading about Dr. Markowitz's sort of pre-IARPA days, go into PubMed, check out some of his peer-reviewed literature on these areas, and you see, as you were just saying about the neuromorphic computing, you see that happening even when you go to your dissertation, which is online, and you're publishing on these local field potential oscillations. Bing, neuromorphic computing one day. And yeah, I mean, it's also fascinating. You see the bridge in action. So I recommend all our audience to go check out the early work as well, because it is quite fascinating.
Thank you, Ira. And you are the first interviewer who has ever encouraged their audience to read my dissertation.
I think they should, because no, it's really a great story, and you see the journey in action. So I mean, it's spot on. Before we get into your portfolio, I'd like to do sort of a little bit more of a deep dive into IARPA. You go to the website, boom, the first thing that comes up, 4 words in bold: high risk, high payoff. And we've heard this theme. We've had the honor of having DARPA guest on the show, working in some similar areas over the last couple of years. Dr. Jeffrey Ling, who recently, he left the Biologics Technology Office over there. When I had him on, he was talking, we do things differently at DARPA. We're not like a venture capital organization or a traditional funding agency. We don't spend 9 months doing due diligence to figure out why not to do something. We just find really cool things to do and put a lot of money behind them. Introduce us a little further to IARPA, a little bit about where sort of the directives come on a daily basis. And then, we also heard a little bit about sort of DARPA, program managers, they spend a certain amount of time there, but then move on. Are there similar models at play in IARPA? Are you allowed to hang out a bit longer to execute on some of these cutting edge tools and technologies? Take us a little bit on that path, if you would.
Happy to. Well, the ARPA model writ large is fundamentally different from your traditional grant-based funding model in government or in the philanthropic domain. ARPAs have customers. We have constituencies that we serve. And our missions are to deliver impact for a particular constituency. So fundamentally, everything is driven by a need to solve a problem, to improve on how work is done, or to enable something that wasn't even possible previously. At IARPA, I mean, we were modeled after pre-existing ARPAs. DARPA is our sort of older sibling. Our constituency is the intelligence community, which has a particular problem space that we support, which ranges from the analysis of all the data that the intelligence community collects to the collection of the data in the first place. And computing is a huge part of both of those, and increasingly biotechnology is a huge part of both of those. So the operating model of IARPA is, as you said, based on high risk, high reward. We're not in the business of delivering incremental advances. Everything we do has to have a quantifiable 10 to 1,000x improvement over the state of the art.
Okay.
And at IARPA, we are rigorously quantitative about how we set goals, how we evaluate progress during the execution of programs, and how we measure impact. Different ARPAs differ in their how much emphasis they put on those quantitative goals, and in particular on tracking performance. So at IARPA, we spend about 20% of every program's budget on independent testing and evaluation, which means we, the government, contract with science and technology experts in a particular field to serve as referees, essentially, in real time, where they are exposed to work that's mid-execution. They take delivery of work products, whether it's hardware, software, chemical reagents, and they independently analyze it to confirm that the progress that a particular research team asserts they are making is actually being made. And that actively informs our funding decisions that are made at roughly 6-month intervals during program execution. Some of the ARPAs don't put as much emphasis as we do on independent testing and evaluation. It's actually a big time sink for the program management staff, but it's key to our ability to deliver impact for our customers. I think once you reach a certain scale, once you reach like a DARPA scale, where you've got upwards of, well, I don't know exactly how many research programs they have, but let's say somewhere between 500 and 1,000, maybe it's not as feasible to invest as much of your time on independent testing and evaluation. But IARPA is a newer research organization. We just celebrated our 15th anniversary. And at the scale that we're operating at currently, we are still able to invest a lot of our time and attention in independent testing and evaluation. And we have the receipts that prove that that leads to real impact for our customers. Our model is to actively manage research. So I began by contrasting this with your traditional grant-making model. And there's a place for every funding model in the science ecosystem. More money for science is great, no matter where it comes from. In a traditional grant-making agency, there is a kind of a broadly scoped call for proposals in a particular area. And you have a study section of peers that are typically drawn from academia, maybe 20 people who will review all the proposals and score them. And then that is what determines who gets funded. There's a certain pay line, right? And there's, although, taking NIH as a model, although like an annual progress report is required, there's no real accountability to achieve progress toward a specific goal. In fact, one of the features of grants is that sometimes you discover things that you weren't expecting. Sometimes you move off in a new direction because you learn something mid-execution. The IARPA model is different from that. We have narrowly scoped programs. So we'll put out a solicitation that says IARPA wants to solve problem X. And we believe that the general category of approaches that seem best suited to solve that problem are X, Y, and Z. And so we're looking for proposals that come from this ecosystem of approaches. And the way we're going to measure progress and the points in time at which we're going to measure progress are defined in this schedule, right? So we require proposals to demonstrate particular quantitatively assessed progress on a schedule. Now, one of the most fundamental things that I always say to my friends and my family is you can't do science on a schedule. Right? You can't just plan to make a big discovery 6 months from now. IARPA's programs, we do a lot of research before we launch a program. And so we are specifically calibrating our program design to achieve advances that we believe are doable on a schedule. We're not just saying like collect a lot of data and then solve the brain within a year. That would be ridiculous. But we can say, we want you to build a device that has a particular set of performance characteristics by 12 months into the program, right? And then maybe we'll have some more open-ended goals about how you use the device after that, which are more sciencey. So IARPA program managers actively manage the execution of the work. We put out a solicitation, we receive proposals, we competitively evaluate them against the requirements of the solicitation. They're not evaluated against each other, like you sometimes see in an academic study section.
Yep.
And then we award contracts. So IARPA does not award grants. We award research contracts under a particular authority that we have under the federal acquisition regulations. So these contracts are negotiated contracts between the government and a particular research team where they put themselves on the hook to deliver certain things and to meet certain milestones. And the program management team's responsibility is to work hand in glove with the performers, is what we call them, the contractors, during execution, to track progress, to help troubleshoot problems. We don't direct the day-to-day of the work. That's all specified in the contract, what they're going to do. But we are empowered to be in the trenches through site visits, through status reports and regular meetings, technical exchange meetings in person. And that affords opportunities for the government to add value throughout program execution. It's clear, if it's clear that a particular approach isn't working and we need to pivot, I'm empowered as a program manager to help make the contractual changes that allow us to do that. If it's clear that we need more resources or not just money, but I need to provide equipment or facilitate the right kind of partnerships to help a project succeed. I'm empowered to do that. And that's actually been instrumental in the success of several of my programs. There is no IARPA program where you just sign the contract and then just set things off to run, and 4 years or 5 years in the future, either it succeeded or it didn't, right? We actively manage our research programs. It's a feature of our model. And that's a feature of the ARPA model in general. So that is why it's imperative that program managers at IARPA and at our sister agencies be highly experienced scientists and or technologists, and ideally have experience leading teams. You can't walk into running an IARPA program having never run a large-scale ambitious research project before. Sure. This is not a place, although IARPA does empower us with the tools to improve as we are doing the job. And I have, being an IARPA program manager has been a masterclass in how to lead high-risk, high-payoff research and development. But the most successful PMs, in my experience, are ones who come with many of the tools that they need in order to cultivate and support and to lead multidisciplinary communities who are pursuing goals that nobody thought, and maybe people still don't think, are possible, right? There's a certain, I don't want to say arrogance, but there's a certain self-confidence that you need to have in order to fully embrace the ethos of being an IARPA program manager. And that comes from having tried, failed, and then having had the courage to try again. I think having experienced failure is necessary in order to be successful as a PM at IARPA. So I think I hit it at all of the questions that you posed. Let me know if there's anything where I could go into greater detail.
No, that was spot on. I definitely appreciate that overview, David. That was great. And I really wanted to contrast, some of what we heard from the DARPA folks, and that was perfect. So I appreciate laying that groundwork. So I would love to segue into some of your portfolio. I think a great place to start would be FELIX. And again, for those listening, Dr. Markowitz is going to give us some top line info here. There is a really cool presentation on YouTube. Actually, it's a meeting, a discussion group between IARPA, Ginkgo Bioworks, and Draper, who are the partners in this particular program. And this is about a 2-hour discussion presentation on all these technologies. I encourage anyone that's interested to go check that video out. But here we have FELIX, again, focused on revolutionizing biosurveillance and finding, as you talked in this other presentation, finding engineering sort of in samples somewhere, complex samples, biologic samples. And again, here is a topic that, we've touched on from different angles with some of our other guests. Andrew Hebler from the White House joined us a couple of weeks ago talking about gain of function, and, good things can happen there, but bad people can do bad things, and we need to know about it. At the same point, once again, thinking about dual use and industry, if I create that frost-resistant orange or the asparagus that grows in the Sahara Desert, I want to know that I got some patents on those technology. I want to know if someone's taking them and using them. Talk a little bit about biosurveillance, both from sort of a national security perspective as well as industrial applications, and then a little bit about sort of the history of FELIX.
Happy to do so. The biotechnology industry has developed impressive new capabilities over the last 10 years that have empowered us to make edits to the genome of basically any organism.
Yep.
And the tools for genetic engineering have become progressively easier to use and more widely accessible, to the point where now your average novice biohacker, which is a really cool hobby, by the way, has the ability to engineer plants, insects, bacteria, what have you. So this is hugely beneficial to society. It's, having easier to use bioengineering tools makes it easier for us to do research on the critical path to developing improved therapeutics, improved agricultural products, improved biomaterials, right? It's revolutionizing the economy. But when you have tools for genetic engineering that are more broadly accessible, it potentially changes the threat landscape as well. The potential for accidental or deliberate misuse of genetically engineered organisms increases along with the accessibility of these tools. If you're the intelligence community, our mission is to stay abreast of evolving threats in the world before they cause problems. Yeah. And so biosurveillance and biodetection of engineered organisms is a major priority for the intelligence community, for domestic law enforcement, right, as well. Now, historically, the tools of biosurveillance were a little bit limited and largely manual, right? Before we did the FELIX program at IARPA, tools for detecting genetic engineering were mostly focused on plants and on just a few plants, and the protocols were largely manual. So they required a human subject matter expert in the loop at every step. Mm-hmm. These were not approaches that scale. These were not approaches, to handle lots and lots of samples in a short span of time. And of course, you lacked that breadth of being able to look for engineering in any organism. And that's the motivation for the FELIX program, is to empower the national security community with new tools that free us from the encumbrance of human manual labor, right? Is to give us tools that allow us to rapidly screen for and detect any signature of engineering in any biological sample at scale and in operationally challenging samples. We scoped the program to have two what are called technical areas. One is focused on experimental platform development, and one was computational platform development. The experimental platforms were focused on delivering new experimental assays that would allow us to rapidly detect things in the field. One of the tools that we developed on the FELIX program is an experimental platform that makes it easy for intelligence professionals and law enforcement in the field to rapidly screen a biological sample, any biological sample, for signatures of engineering. And it's called a miniature microarray, and I will hold it up here.
There you go.
All right.
Very cool.
It's a tiny chip that's about the size of your thumbnail that has tens of thousands of tiny DNA probes mounted on it. And all you do is you incubate, there's a particular protocol for preparing your samples, but you incubate your biological sample of interest in solution with this microarray for a minute or two. And then there is a device that allows you to rapidly read out with a fluorescent readout on this microarray whether there is a signature of interest in the sample. We also have the ability to capture suspicious DNA that bears the hallmarks of engineering, and we can then remove that from this chip and use it for downstream bioinformatic analysis. So you can actually look at the sequenced DNA of interest and the surrounding context in a bioinformatic platform. So that's one of the tools we developed through FELIX. And so that allows, if you're law enforcement and you are screening for intellectual property theft at a port, right? Somebody's trying to smuggle some vital technology from the US bioeconomy out of the country, we can probe for that quickly with these sorts of arrays. It can also be used to identify pathogens that shouldn't be where we expect them to be, and other things. FELIX also developed a suite of computational platforms that use machine learning to rapidly analyze very large-scale genetic sequencing data for suspicious signatures of engineering. So this isn't like a traditional database lookup approach where like you know exactly what to look for and then you just do a find and match type of analysis. The beauty of machine learning is it's a statistical approach. So with large enough training data sets of engineered organisms, we can train machine learning models to learn the statistics of when one thing doesn't belong in a sample where it's found. Maybe there's a foreign insert from one organism's genome into another organism's genome from a totally different species. Maybe there's something that is similar to a type of insertion or deletion or other modification that we've seen previously, but it's not identical. So machine learning systems afford us the ability to generalize that detection capability to novel forms of engineering that haven't been seen before. And as we discussed during the press conference that you referenced earlier that IARPA hosted with a couple of our performers, with Draper Labs and Ginkgo Bioworks, these tools are able to detect signatures of engineering at about 70% sensitivity and nearly 100% specificity, meaning there's almost no false positives. It's like one false positive we got on the program.
Amazing.
So it's really been remarkable to see the progress that FELIX has achieved. We now have tools that allow our customers in the national security community to screen thousands and thousands of samples with computers automatically, where a human would have previously needed to sift through all the sequencing data. And now these systems will flag for human analysts which samples are of greatest interest and worthy of their limited time to go and study an engineered organism and try and assess the threat level. What's really exciting about the FELIX program is we have put these tools in the hands of customers around the world now. And that was one of the things that we were really proud to announce at the press conference that was held about a month ago now, at the time of recording of this podcast. So IARPA has delivered these experimental and computational biodetection tools to double-digit numbers of customers throughout the United States national security community, as well as our international partner community. So these tools are being used and they will be keeping all of us safe in the future. And so that's an example of the kind of impact that we use to measure the success of IARPA programs, right? It's not a financial impact. You can measure it in terms of lives saved. And it's been really gratifying for me as a PM.
Awesome. Really awesome. David, moving from FELIX to MICrONS, because now here, once again, coming back to your dissertation and all the work you did in field potential oscillations and spiking, and obviously the segue to what this potentially could be in terms of designing various machine learning, artificial intelligence, what have you, opportunities to act, perform like what happens in here. Take us a little bit on, because obviously this is maybe a little bit more long-term than, or maybe not, you can tell us, but neuromorphic computing is a theme that some of our guests have spoken about. Christoph Koch a couple of months ago was on talk, but he was more interested whether neuromorphic computing could be conscious at some point, but we'll leave that for another day. Talk a little bit about where you see work like this going, because clearly, you got some of these quantum computing folks. We'll keep them in this room over here doing their thing, but designing the future of computers based on this amazing evolutionary perfected computer is a whole other story. Take us a little bit down this path and on what you foresee this ultimately leads to.
It's a great question. I actually just gave an invited keynote talk on this at the supercomputing annual meeting in Texas. It's a meeting with like 12,000 people. And I got up and I gave a talk called Biology is All You Need, which really took some gall to get up in front of a room full of high performance computing experts and say, you should all be looking to biology. But let me set the stage for this this portion of the discussion
Sure.
and talk about a major pain point in high performance computing today, which is scalability and power efficiency. If you look at the largest AI systems, right? So we're in a golden age of AI. In the last 10 years, we've seen massive improvements in the capabilities of computer vision systems, of control systems, of language models. And the leading edge of that is always the largest model year over year. Whichever model has the largest number of trainable parameters, that's a pretty good proxy for the size of the most capable models. And so they've grown from a few thousand trainable parameters in 2012, 2013 to the largest model in 2022 has 540 billion trainable parameters.
Yeah.
It required over 6,000 custom chips powered by megawatts of power, training for days and days in a data center. And it required billions of petaflops of compute power to train this model. I mean, the resource requirements and the carbon footprint of today's largest leading edge AI systems is becoming a real problem. And if you think forward to a future, and it's my job as an IARPA program manager is to think about what the future is going to look like. If you think forward to where we want to train AI systems that have 2 or 3 orders of magnitude more trainable parameters, 100 trillion parameters would be about 1,000x.
Yep.
It's prohibitive. The cost of training one of those models in the commercial cloud would be tens of billions of dollars. The carbon footprint would be equivalent to about 200,000 US homes, the size of a medium-sized city. We just don't have a scaling path with today's compute hardware and training paradigms for AI systems, for machine learning systems. And so biology has already solved this problem for us. The biological wetware between our ears operates a more than 100 trillion parameter model for less than 20 watts of power. And the cost of training a human brain 24 hours a day for the first 18 years of life is equivalent to operating about 1/3 of a US home for 1 year. So, which is like 8 orders of magnitude lower than the cost of training a 100 trillion parameter AI system in the future. So there's a fundamental need for innovation, both in hardware and in the algorithms that we use for training machine learning systems. And I just wanted to set the table there.
Okay.
Of course, because biology has solved this problem, IARPA's hypothesis is we need to be looking more toward the operating principles of the brain in order to delineate clear guidance for computing architecture development and for machine learning algorithm development in the future. Neuromorphic computing, right? We're in a golden age of neuromorphic computing, where in the last 10 years, we have seen also a proliferation of ever more capable platforms that improve on the power efficiency of conventional GPUs by several orders of magnitude, but they're still, a million-fold off in power efficiency from regular brains. Even though neuromorphic platforms use spiking, event-based communication, and they use other tricks to minimize the amount of data that's moved around. So there's still some fundamental disconnect between the operating principles of today's neuromorphic platforms and the biological brain.
Yep.
That is the motivation for the MICrONS program, is to just look at the thing, right?
Yep.
Let's develop the tools that we need to develop a really detailed computational understanding of neural circuits with a focus on cortex. As I said before, that's the evolutionarily newest part of the human brain, of the mammalian brain. It's the wrinkly part on the outside. So on the MICrONS program, we targeted the development of new tools that would scale up to allow us to map the synaptic connectivity and the activity of neurons at the cubic millimeter scale. Now, that sounds really tiny, but for a mammalian brain, that is sufficient scale to encompass multiple brain areas that have different functions. And it's big enough to let us look across all of the depths of cortex, from the pia and layer 1 all the way down to the white matter that comes out of layer 6, where a lot of axons exit the cortex to go to their long-range targets. So the MICrONS program was focused on developing the full technology stack needed, the experimental tools and the analysis tools needed to gain that multimodal understanding of the brain at this unprecedented scale, which was 3 orders of magnitude larger than what anybody has been able to map before this program. And we succeeded in generating the largest single biomedical imaging data set that's ever been collected. So 1.4 petabytes of data. This encompasses the structure, the synaptic connectivity, and the responses of about 100,000 neurons in a cubic millimeter volume. As soon as we had those data, we made them publicly available, because this is such a singularly valuable data set, not just for computing and machine learning algorithm development, but for biomedical applications as well.
Sure.
We wanted to build a community around the analysis of these data as quickly as possible. And so through generous partnerships with several cloud computing providers, Google Cloud and Amazon Web Services, we freely shared the data. And you can go in your web browser today to microns-explorer.org and visualize these data in your web browser for free. We also have an API that allows researchers to programmatically interact with the data to do real detailed scientific analyses. And this rapidly became one of the most actively used public data resources in neuroscience, which is really gratifying for me as the program manager to see this have impact for a lot of laboratories around the world. But in terms of what value we think this is going to have for neuromorphic computing and machine learning, which I think was your original question, we're already learning some fundamental new operating principles about the brain that have immediate bearing on machine learning. So one of the most fundamental questions we'd like to answer is, what learning algorithm does the brain use? I mean, we know about the learning rules at individual synapses, but algorithmically, like, it's still a mystery. If you look at the predictions made by the predominant learning models in the machine learning literature, they use something called backpropagation through gradient descent. And the prediction of those models is, if you look at the weights of synapses in these models, they should have a continuous graded analog distribution with a single mode, like a Gaussian distribution, okay? Some strong weights, some weak weights, and then some distribution in between. When we look at the size of the synapses, at the volume of the postsynaptic terminals in MICrONS data, which is highly correlated with the strength of synapses that's been established through physiology studies, we don't see that single analog distribution. We actually see what looks like a sum of a binary variable, because you see a sort of a multimodal, almost binary distribution of synapse sizes, combined with an analog variable that is log normally distributed. So this, for the first time, provides a very clear constraint for people who develop algorithms, because now we know what the final result of whatever learning algorithm the brain uses should look like. It's the sum of these two variables.
Got it.
And it could arise because the brain combines multiple learning rules. It could be that there is a single learning rule that somehow gives rise to these two things. Now we have grist for the mill, like we have guidance to constrain computational and experimental studies in the future. There are a variety of other insights about the canonical microcircuits that we have identified in MICrONS data. So we're learning how, if you look in the brain, and we have been studying the mouse brain on the MICrONS program, there are, there's like 50 different cell types as identified by their morphology, right? The shape of the neurons, how they connect with partners, and then their molecular profile. So when we look at how the excitatory neurons in the brain receive inputs from different cell types, there is a very specific wiring logic. There are some neurons that only inhibit the axon initial segment of a neuron. This is a cell body, my fist here, and this is the axon. You know, there's one class of neuron that only exists to turn off the outputs from its excitatory targets. They know you can't send messages to anybody. There are other types of cells that will only inhibit the apical dendrites, which are the topmost compartment of cells, or the basal dendrites, which are sort of around the cell body. And so we're beginning to delineate the full spectrum of computations that are implemented locally on single neurons, so that we can define what is the minimal computational unit that the brain uses, and that we may benefit from mimicking in machine learning models and ultimately in neuromorphic hardware. One of the reasons why today's computing hardware, and I'll conclude with this, one of the reasons why today's computing hardware is not nearly as efficient as the brain, it's because you have to move so much data around to support computation. But what if we could define the minimal computing unit of the brain in silico, so that you don't have to move data around? Everything is kind of self-contained, and you can maximize the amount of local computation that you can perform and local data storage you can perform. Maybe that's one key to achieving the sort of energy efficiency of the brain. So expect more advances over the coming 5, 10, 15 years. But these are some of the really exciting things we can do today using the MICrONS data that have been generated through this IARPA program.
Outstanding, really outstanding. David, for those that are going to become followers of you, following this show and checking you out in some of these presentations and press releases and so forth that you've given in the past, one of the ones they're going to find is from about 5 years ago, when you were basically running the IARPA Proposers Day for the MIST, the Molecular Information Storage program. Happy to have you talk about MIST as well. I don't want to take tremendous amounts of your time because you got more important things to do. But if you could say a few words about how Proposers Day work. I mean, I know this is a really unique function of the ARPAs in the sense that, you help lay the groundwork for all sorts, whether they're big companies or small startup innovators that want to get involved and work with these programs. And then also, if you could say a couple words about the Seedlings program, which is somewhat analogous, but it works alongside. That'd be great too.
Happy to. This gets into some of the mechanics of how IARPA does business.
Yeah.
Right. Every IARPA program starts with an idea and a person. We rely on smart, motivated people to come to IARPA and say, I have a great idea for a program. I would like to do this program at IARPA. Although there's a lot of overarching strategy because we know what the priorities are of our customer group, at the end of the day, if you don't have somebody who's really passionate and capable about solving a particular problem, like what's the point?
Right.
So once we have a PM that has come on board and is interested in delivering new capabilities in a certain area, then the work begins of designing the program, putting out a solicitation, and then doing community engagement. And we rely a lot on workshops and ultimately proposers days to do that. So I've organized workshops at IARPA that have nucleated entirely new fields by bridging biology and engineering and computing. The DNA data storage program that I run, MIST, helped create an entirely new industry through several workshops. And we use proposers days as kind of the final workshop before we receive proposals. And so this is an opportunity to engage directly with the community in person, tell them exactly what the program looks like, what our goals are going to be, and to provide opportunity for the community to give the government feedback on what is good about the program design, what could be better, and what they think is the right sort of approach to address a certain problem. So proposers days can really catalyze new collaborations that didn't exist before. We get a lot of participation from industry, from academia, from nonprofits as well, to build the right coalitions. And on the MIST program, which is the DNA data storage program that I run, our teams that we ultimately awarded contracts to are made up of exactly those types of coalitions, where you have an academic prime contractor that has particular experience with systems integration and research management, partnering with industry, where you've got startup companies or well-established device makers, as well as other academic institutions. So on the MIST program, one of our collaborations is between Georgia Tech Research Institute and then several startups, Twist Bioscience and Roswell Biotechnologies and University of Washington, and Microsoft is also a participant in that team. So we really, we create opportunities for collaboration. We align incentives between these different groups so that a company can build a product that's on the critical path for a particular commercial roadmap, and they can retain the rights to build and market a particular product. But the process of developing that capability leads to the government getting something that we want. So hopefully that tells you a little bit about the how we use Proposers Days. And I'll just mention that when we hosted that Proposers Day for the Molecular Information Storage, or MIST program, several years ago, the vision was we were going to deliver new technologies for writing massive amounts of data to DNA at scale. And this is the, I mean, you can see yourself in the reflection here. So this is a next generation DNA synthesis chip that can write several gigabytes of data to DNA. This is several orders of magnitude smaller, and it has several orders of magnitude higher throughput for several orders of magnitude lower cost of DNA synthesis than any technology that existed before the MIST program. And so that's an example of some fundamental new technology that IARPA can develop through our programs. And it all starts with the Proposers Day.
Awesome. David, your thoughts on how we encourage more David Markowitzes. You mentioned at the beginning, you obviously got to come to this with this. We have to encourage more of this convergent thinking. And we don't just study biology. We don't just study computer science. We study the combination of these different disciplines, and it's the bridging, the intersection where we find the sort of the quantum leaps forward. How do we get more David Markowitzes to think convergently? How do we encourage our institutions to create them? And at the same time, how do we get them to IARPA and away from that Musk character at Neuralink or out of the pharmaceutical industry? Give me some thoughts on this aspect of it, because we need that next generation to, we have 100 David Markowitzes, we have no more problems in the world. Where do we go?
That is a $1 trillion question, Ira, because honestly, if we had hundreds more people who were empowered to attack some of our nation's hardest problems with the resources that I'm fortunate to have available as an IARPA program manager, we could deliver almost limitless impact and make an entirely new and vibrant economy, right, for America in the future.
Yeah.
I want to answer your question first by acknowledging my privilege. Is that I am privileged to have grown up in a home with very highly educated parents who told me from a young age that I could do anything, and that failure is okay, and that they would support me, through my failures, no matter what. And so that created a mindset where I felt empowered from a young age to chase whatever problems interested me, to try and fail. And I failed in a lot of things during my academic career. And to be able to get up, dust myself off, and just keep going and trying hard things and risking failure. it's a cultivated mindset. And I recognize that I'm privileged to have been brought up with that mindset, where a lot of people aren't. And I think that it's a sociological problem, but in terms of how we can cultivate more people who have that feeling of empowerment to go and follow their interests wherever they may lead, and who aren't afraid of failure. From an educational standpoint, I think that's a mindset that schools can try and instill in students, particularly at the university level. The sort of multidisciplinary background that I cultivated, I kind of had to cobble together myself. I think that in neuroscience and in biology, we have started to see more of a proliferation of what are called integrative training programs, where you're not just focused on biology, but also on quantitative modeling, right? So you're bridging some of the informatics and the hard science components of these fields. For me, I always had the itch to build new things, to solve problems. I don't want to say at the fringes of how they're solved today, but you know, I'm excited by discovering a new way of doing things. And for a certain type of student, I think that having opportunities for them to get involved in research from a very early age can be instrumental for putting somebody on the path where they can ultimately become a program manager at a high-risk, high-reward research agency. So I benefited significantly from fellowship training by the US government, which specifically encouraged research at the interface between high-performance computing and hard science fields, right? And that's I think today that fellowship program may have like 50 people who are funded by it in the entire United States. They may only give awards to like 1 or 2% of applicants. So more funding for training programs that empower people to work at the interface between fields, to take risks and fail knowing that their funding for their graduate degree or their postdoctoral work is secured. Having that safety net, right, to give everybody that sort of privilege that I have enjoyed throughout my career, I think could make a huge difference. And ultimately, that will create a funnel that will benefit IARPA, DARPA, and all of our sister agencies. Will give us the excellent people that we need solving our nation's hardest problems.
Excellent message. Really excellent message. David, while we have you, anything else coming up as we get ready to enter 2023? Any upcoming proposers days that we should know about? Conferences that you're going to be presenting at, places where we can listen to you, potentially meet you face to face at some point? Anything else that I missed that you want to mention? Please have the floor.
I appreciate the opportunity to do that. I just, like I said, just gave a keynote talk at the Supercomputing annual meeting a week before this podcast was recorded. So that's my major public engagement for a while. IARPA recently launched several new programs in high performance computing and in biosecurity. We don't typically announce new programs before we announce a proposers day. So I can't telegraph that, you know, IARPA is going to be putting lots of money into Area X next year. So everybody start getting your ideas ready.
Gotcha.
But I do think that biology and problems at the interface between biology and other disciplines are occupying increasing mind share in the national security community. And we want the best people around the world helping us solve problems in that domain.
Wonderful. Really wonderful. It was awesome listening to David. I'm amazed by the scope of your work, the depth of your portfolio, and rooting you on, rooting you and everyone at IARPA on for everything you're doing to move this area, all these areas forward, and at the same time to protect the country. Again, for everybody that is going to be listening to this particular episode across the various podcast networks or watching on our YouTube channel, you've been listening to the fascinating Dr. David Markowitz, Program Manager at the Intelligence Advanced Research Project Activity, or IARPA, doing amazing high-risk, high-payoff research. David, I want to thank you again for taking the time out of your schedule to come educate us on all these topics for a little while. Obviously, thank you for doing them and keeping our country safe. And as we like to say on our show, thanks for helping to create a better tomorrow via what you do. Really such an inspiring story.
Thank you, Ira, and thanks to your listeners for your time. It's been a privilege.